This is the lecture for Lab 5, Differential Stains. The purpose of this lab is first to understand the purpose of differential stains, second to understand the process of a gram stain and the reagents used, and lastly to correctly perform a gram stain. When performing a differential stain, there are several different types of reagents that are used. The first one that is usually used is a primary stain. And the primary stain is going to identify a specific component or a specific structure in the bacterial cell. The second reagent that would be used is a mordant, and the mordant is going to enhance the primary stain. The third reagent that would be used is a selective treatment, and the selective treatment is going to show a difference in cells that have the structure and cells that do not have the structure. And often the selective treatment can be heat, adding heat to the samples, or adding alcohol. And then the third reagent that is often used in a differential stain is a counter stain. And the counter stain provides a contrast to the primary stain. And in a gram stain, which is what we will be focusing on in this lab, the primary stain is the crystal violet, the mordant is iodine, the selective treatment is alcohol, and the counter stain is safranin. The gram stain is used to identify bacterial cells by their cell wall type. And we actually refer to the cell walls as either gram-positive cell walls or gram-negative cell walls. And again, the difference is in the structure of the cell walls. In a gram-positive cell wall, there is a very, very thick layer of peptidoglycan. In a gram-negative cell wall, there is a very thin layer of peptidoglycan, but those cells also have an outer membrane, which is a phospholipid bilayer and contains lipopolysaccharides. So the process of the gram stain is allowing you to differentiate between the two types of cell walls. When performing the gram stain method, the first thing you will do is prepare a smear. So a smear, remember, is a sample of bacteria on a slide because you will be observing the individual cells and whether they are gram-positive or gram-negative on a slide. Now for the gram stain, what you want to do is actually have 3 different samples. So you are actually going to create 3 smears on one slide. And you may want to use the wax pencil to draw on the bottom of the slide 3 different sections so that you don't mix the bacteria. On one side of the slide, you want to make a smear of a gram-positive, a known gram-positive. On the other side, you want to make a smear of a known gram-negative. And on the middle is where you have your unknown sample. And this is because in the gram stain, there are different places where you can make mistakes. So you want to have a positive control and a negative control so that you can trust whatever the results are on your unknown. For the process of the gram stain, this is a diagram or a figure that shows that. First, you are going to begin with your heat-fixed smear, and again, 3 samples with a gram-positive, a gram-negative, and an unknown. Then you will cover the smear with crystal violet and let it stain or sit on the cells staining for 1 minute. After that, you will rinse with water. After you have rinsed with water, you will add iodine, so that's the mordant, for 1 minute. After you add the iodine, again rinse with water. So you'll rinse with water, and then you add the selective treatment. And for the selective treatment, this is ethanol. So you're going to add alcohol, and you're going to let the alcohol flow over the slide, and you rinse the slide with alcohol until it runs clear. So that's not going to be a very long time. And this is a very important step. You want to make sure that you don't add too much alcohol or you don't add too little alcohol. After that, you would rinse the slide with water, and that's very important to rinse the slide with water once the alcohol has been running clear. After that, you stain with safranin, the counter stain, for 1 minute, and then you are finished. So then you can blot dry the slide and you can observe under oil immersion and determine what the gram reaction of your unknown is. For the results, if your cells are gram positive, meaning that they have that very thick layer of peptidoglycan, the cells should appear a dark purple or sometimes blue depending on the intensity of the light. Gram negatives should appear pink or red, again depending on the intensity of the light. If you see the following result, where you see both purple and pink cells, that is a mistake. So you have a mistake in the procedure, because all cells of a particular species should be all 1 gram reaction. So if you see something like this, that means there is something wrong in your procedure and you need to repeat the gram stain. You should be aware that the gram stain is not the only differential stain that is used. One of the other more common ones is called the acid-fast stain, and this is used to identify members of the Mycobacteria genus. And Mycobacteria leprae causes leprosy, Mycobacteria tuberculosis causes tuberculosis. And the reason you use an acid-fast stain is because these bacteria have a lot of waxes and mycolic acid in their cell wall, so a gram stain does not work well on them. So if you need to identify members of the mycobacteria, you need to perform an acid-fast stain. And just so you are aware of the procedure, it also involves a primary stain, it also involves a mordant, and then a selective treatment, which is alcohol, and then also counter stain. And a positive color for the acid-fast stain would be if the bacteria turn pink. So this is a stain looking for Mycobacteria tuberculosis. And so all of these bacteria belong to that group.